COVID-19 has caused panic around the world for right reasons. It is caused by a virus called SARS coronavirus 2. SARS coronavirus 2, commonly known as coronavirus, is highly infectious. Some people doubt that this virus was made in a lab as biological weapon. While we may never know for sure whether the virus was created in some lab or not, we must understand that through natural evolution, new viruses may emerge from old ones. So here, using coronavirus 2 as example, we are gonna explore where do novel viruses come from. COVID-19 virus or SARS coronavirus 2 spreads through air. Like older coronavirus family members, it primarily infects respiratory tract. Here it enters in the ciliated epithelial cells lining the respiratory tubes. Let's now look into one of the epithelial cells which has been infected by the virus. One objective of all organisms, including viruses, is to ensure survival of their kind by reproduction. At the end of each infection cycle, many new viruses are created. This is achieved by using information brought in by the parent virus. But some of the new viruses may not exactly match the parent virus. Therefore, among the virus offsprings, there may be some which almost exactly match with the parent virus
and others that don't.
But how does this happen? To understand this, let's see the virus structure more closely. If we could just cut open the virus and look more closely inside, a string-like structure is revealed, which is composed of proteins and genome. The proteins shown in yellow protect the genome from damages. If other virus coatings and proteins are washed off, genome is stretched out. And looked more closely, it looks like a necklace with pearls of 4 different colors: blue, green, red and yellow. Each pearl is called a nucleotide. Unique arrangement of these nucleotides serve to carry unique set of information to build unique structures. Just as we can differentiate between different English words because of unique letter sets that make the word, cell can understand and act upon information written in the language of nucleotides. To elaborate it further, same alphabets DENOORW repeated same number of times, but when put in different arrangements make different words. New door in one arrangement and one word in another. In the same manner, 4 nucleotides in different arrangements of sequences can make different structures. All organisms, including viruses, need to carry genome inside them in order to make their copies or produce their children. Genome of COVID-19 virus has some 30,000 such pearls or nucleotides. Each new virus particle has to carry a copy of its genome before leaving the host cell. Therefore, several copies of virus genome must be produced in each infection cycle, which are threaded inside new viruses at the time of their production in the cell. Production of as many genome copies as there are virus particles is achieved through the process of replication. But the process of replication is never accurate. For corona-like viruses, on average, one mistake is made while copying 1000 nucleotides. If you pay attention in this example, each copy has some wrong nucleotides added to it. But these mistakes are ultimately responsible for the emergence of new virus types. Let's zoom out of these genomes in this example. Each differently colored string represents slightly different version of the genome, which results from errors in the process of replication. So far, we have learned that genome carries information to build new viruses. When copies are similar to the parent, new viruses resemble the original virus. But when replicated genome differs significantly from the parent, new potential viruses emerge. Among all the virus offsprings, this virus matches with the parent virus, therefore it has same infection characteristics as its parent. But these 3 types are different because they carry changed genome. These changed viruses are called quasi-species. Among them, those who would have chance to find and infect new hosts would emerge as new species. Therefore, SARS coronavirus 2 may have emerged through the significant changes in genome of original parent virus, which originally had a different host. Scientists have hypothesized that original virus or its copies could infect bats or civet cats. but one of the quasi-species was able to infect new host, in this case humans. Summarizing the above discussion, original virus is the parasite of bats or civet cats. One of the successful quasi-species is now called SARS coronavirus 2, which is the cause of current pandemic. All other quasi-species may not be fit enough to infect their previous host or any prospective host. Therefore, they would die off. With the basic understanding of how viruses may change during the course of an infection cycle, let's now have a small discussion on whether the COVID-19 virus was created in some lab as a bioweapon project or not. From the above discussion, we now know that new species of virus may emerge naturally. Here is the catch, however. Processes that nature uses to change viruses can also be used in a lab setting to change organisms such as viruses. You may get surprised to know that virologists are creating new viruses all the time. Intention of creating changed viruses, however, is to understand the properties of natural disease causing viruses. Okay, not always, only most of the time. There are hundreds of very important reasons to change viruses or engineer viruses in a lab. That if I am asked whether COVID-19 virus was created in a lab as bioweapon or did it emerge naturally, I would say that there are more chances that it emerged naturally. Nonetheless, scientists keep creating new viruses and it is for our own benefits. Do you wanna know what are those benefits? Subscribe my channel and hit the bell icon to know the answer in my future videos.